సంతకం కూడా ఉండాలి. ఎలా రాసావు ఎగ్జామ్? అందరికి సేమ్ పేపర్ నానా? కాదు, సెక్షన్స్ గా డివైడ్ చేశాడంట, 25, 25, 25, 25 సపరేట్ సపరేట్ గా. ఎవరు సేమ్ పేపర్ వస్తుంది అందరికి? డిఫరెంట్ పేపర్ ఎందుకు వస్తుంది అందరికి?
R&D projects and has received Innovation Award 2020 as a part of the India Innovation 2020, and he has received Innovation Performance Trust Award 2019 for GSK's R&D's most impactful performance, and he has also a patent regarding the controlled release formulations with Balagram Indian Patent Office granted by Indian Patent Office. And he's at present working in Executive Pharmaceuticals LLP Hyderabad as his director. And he has also guided various PhD students as well as science students for the project. It is my immense pleasure, sir, to welcome you for today's session. Please say yours.
Good afternoon once again to all the faculty uh, who can be there, Dr. Massan and the principal is not here. Convey my regards for the this order. Teaching staff from me, non-teaching staff from me, research scholar from me, as well as other faculty from different organizations and my student friends. So it's a warm welcome to you and I extend my warm wishes to you as well as great performance. So as soon as I received an invite on this topic, like you have seen that next gen, I thought how could I present it to you? And I have seen also on the resource persons, there are very few industrial resource persons. So this presentation is mainly concentrating on the AI tools or AI softwares, AI based softwares, which are used in the industry. Okay. So I might not touch topics on the academic aspects because there are more pioneers here in the list who are from an academic background. So this is purely an industrial list. Apart from this, I think I have two more challenges. Now I think it's almost lunchtime, so I expect it's like energy levels. But I request you to just pay attention. And the second challenge I have is that so I need to break the ice. I think you're all aware of virus. So in virus, what I get is that when the first member goes to the virus, he has all the stress and tension to face that virus. I have similar stress and tension in spite of having not the experience, but still, I try to give. And second thing is, there's one disclaimer which I've not kept on the screen, but I would like to tell you already that these all softwares are being used across the industry and the content has been taken there from their public domains. So I hope that this would not conflict to their rules and regulations. Okay. So what is the current topic on? The current topic is on artificial intelligence, which is driving the pharmaceutical research development. It's a paradigm shift. That is a clear shift. What is happening in the clear shift? We'll go through all the slides. So this is the brief profile. I have a recent experience of 20 years, like which the Kamila has told already. Like I've been in the industry for more than 16 years, and I've done my PhD from Andhra University. So I have close association with Andhra University as well as ANU. Like ANU, I have Dr. Mastan. She is my classmate with my BPharm. So I have that association with her, and I have one more faculty from ANU, which is Dr. Parmir. So he was my junior in BPharm. So and I also visited ANU college in 2021. So I was calculating the number of years. So I have now found that most of the students might have left the organization. So I'm again new to the academy, new again to the students over here. So just a brief introduction, like I am a director or a delivery manager who takes care of end-to-end development of all the products and delivery concepts retaining to my group. Like we take care of right from ideation, innovation to the commercialization. Like for the first time, is the commercial.
Madam, audio is not available. We could not hear.
But still, I would like to say that it's still 90% started, and it is the use of computer. but these are being used as background for pharmaceutical research. Okay, these are few AI ones. Now coming to the drug discovery. Okay, so you might MLP or the financial students might be aware of computer aided discovery.
So just I came to know that it was on mute. So I'll just repeat at least. I'll give the introduction, like what is there and all these things. So just bear with me. So sorry for the glitch. On behalf of ANU, like I may not. I'll just quickly review. Like I was saying that those who are online and who could not hear screen might have seen screen. You might have seen pharmaceutical industry, like the discovery, clinical studies, formulation development, which is my core area of expertise. The supporting departments like quality compliance, regulatory filing, and post-market surveillance. Okay, so artificial intelligence, I think everyone might be aware of
నాకు రాంటి
applications and all
సెట్టింగ్స్
these things.
చూసుకుంటున్నాను
So I've been speaking on that, and also I have spoke about some familiar terms like machine learning, deep learning, and all these things which are on the screen. And these are different commonly explored AI models, like which many of us might not be familiar. But what happens is that these are the background for us to help us in the AI tools or anything like that. And I've just started with the drug discovery. So many of you might be aware of computer-aided drug discovery, which has been very much used in the drug discovery. So this was having accuracy until point of time wherein artificial intelligence has really picked up in the industry. But there are limitations of CAD. Like what happened is that the complexity of biological systems. So we have small molecules and large molecules, the peptides and all these things. So as the size of the molecule increased and as the data has been increased, so there was a lot of complexity of the biological system, which is very much making CAD difficult to help in the drug discovery. But still it is used. There is one disadvantage, that's all. So the difficulties in accurately representing the target flexibility and the limited scope analysis using the traditional methods. So what did AI actually do? What AI does, what it has ability to analyze vast amount of data. Now in the drug discovery, we have huge data. As in my morning introduction note also, I've told that it takes about more than 20 years to develop one molecule, and during these 20 years there are different phases of clinical research, phase 1, phase 2, and all these things. We do some preclinical trials, clinical trials, and all these things. So during all these 20 years there is huge data which is developed. So once using the human intelligence, it might be very difficult to recall the first page. Suppose we read a newspaper, it is very difficult for us to recall what is on the first paper when we reach to the sports column on the last paper, at least sports column or whatever might be the news in the last paper. So what in the 20 years of span wherein they have done the drug development, drug discovery, development, and all these things, what happens is that it would be very difficult to recall the information or to pull up the information, summarize the information. What AI is doing is now is it is taking all the data and using the deep learning and machine learning, it is helping us summarize and give the content which need to be part of the regulatory filings. That is one thing what it is doing. And in 2018, Massachusetts, that is MIT University, has partnered with Novartis and Pfizer to transform the process of drug design and manufacturing with its machine learning and synthesis confirmation. So, seeing the pharma giants, which I have already told you, that Novartis and Pfizer have already started, and in 2018 they applied to a molecule where they have used AI. Now, I'm also saying that, like, I'm covering all the areas, so I might not go deep inside. So I would just give the notes, and you can use the internet to browse and learn more information. So Ceratara AI. So this Ceratara AI, it is a D360 scientific informatics platform in the industry, which is leading solution for small molecule and sequence based modalities research. So this is one of the AI which is clearly used, and when I'm saying these are used, most of them, 90%, I have used in my research development, as well as few are taken from the public domain. But the software which are presented here are majorly I have used. So knowing their use, I have added in the presentation. The second is the Schrodinger, which is used for computational chemistry. But I'm not part of the drug discovery, so this I've just given a basic notes, wherein I'll be covering more in the formulation aspects. Second is on the clinical studies. I was saying that there are a lot of phases in the development, like phase 1, phase 2, phase 3, and phase 4. So during all these course of development in a drug product development, there is huge amount of data developed, and we need to be mindful that by the human intelligence, it would be very difficult to pull the data onto a single sheet or to summarize it in few pages. So what here are few softwares like Pumas AI, which is used by our company. These again bifurcated into Duke Pumas, Pumas CP, and Pumas. And the next is Ceratara AI, which is also used in the drug discovery. Here it is called Codex. There it is called D360. Now one is the Silico screen. All these softwares are being used extensively in the industry, and these have different functions. Like they bring machine learning directly into non-linear mixed effects modeling, combining the interpretability of the mechanistic models and the flexibility of modern AI. So all these terms might be because these have been picked up from the public domain, but in general, if we say that these are helpful in collecting the data, analyzing the data, and they give the summary. So when they give the summary, it would be very helpful for a research scientist to pool the pool of data which comes out as a summary. It will be helpful to predict, because when we give to the regulatory authorities, we do not give 100 or a 500. I'm just giving a number, but it would be lakhs of papers. So when we read on SBOA, which is the NC, that's the new chemical entity literature, when we read, so a scientist is supposed to read that, and it would shift majorly, like the number of pages would be more than 500, 600 for a scientist like me to read it. Now, when they are submitting to regulatory authorities, there should be a summary. It cannot be the raw data which needs to be submitted. So all these Pumas API, Ceratara API, or the Silico screens, all these AI tools in different, they use different algorithms and deep learning and ML to bring this information concise, such a way that the regulatory authorities, it would be easy for them to assess what is the information that has been published, or what is the information that has been submitted by the respective pharmaceutical industry or pharmaceutical companies. So these three are which have been using in the clinical studies. Next, coming to the formulation development, I think this is my core area where I'll be taking a little bit deep inside on what are the softwares which are used. So the flow cycle, like what is the cycle of development which is generally followed in the pharmaceutical industry. Generally, after we get to know the active pharmaceutical ingredient, what we do is that we do the solubility studies, we do the drug excipient compatibility studies, the excipient selection based on that, the impurity screening, followed by the excipient optimization, followed by the packing selection, followed by the stability, manufacturing robustness, scale up and registration matches, wherein we end there to submit a product to the regulatory authorities. So at least I've been using AI in at least 60% in these areas, which practically I've got good output from these AI tools. These are several softwares which have been used directly by our company in the area of pharmaceutical research development. Suppose if I say that literature review, we have Pinjara, which is the pharmacopoeia, and right find expertise. What does this do is that, as I already told you, a molecule which is already, which have been well researched and which is available in the market. So it has n number of pages of n number of literature. Now, when we do a 505 B2 development or a ANDA molecules, what is that is already the FDA regulation itself says that we need to utilize the existing information which is available there in the public domain. Okay. In such case, we need to see multiple papers, like thousands of papers we need to read, which is not possible according to the timelines given to us. So what's the timelines is that within 1 year or 1.5 years, based on the complexity of the project, we generally do the development. We propose the development timelines. And if we keep reading all these thousands of pages, what happens is that more than 6 months we'll be spending on reading the literature. Whereas reading the literature is a continuous process and it is dynamic because throughout the project life cycle, we keep reading literature. But at least to start the project, we need some amount of literature or some amount of physical properties of a drug molecule such a way that into which development the product fits in. Okay. In such case, the Saspingera or find expert enterprise will help us in summarizing or will give us the information which is primarily required for a formulation development scientist, which would help us to kickstart at least the project. Then the second ones, the salt identification, the solubility studies, the drug excipient compatibility. So there are several like Schrodinger, Saspingera, or farm-dense solutions which I have been using in my core development. This LASA limited Zenith, which is used for impurity, drug excipient compatibility, and impurity identification. I think I have one example to show that how it is done exactly. And also on the excipient selections we use in the impurity screening and excipient optimization, the stability of both the drug molecule, the API, as well as the formulation. Even they help in the packing selection. So this is something strange, which for us also that the AI tools are helping in identifying even the packing selection. Generally, packing selection, you might have seen blisters, you might have seen strip packings, you might have seen HDP bottles, you might have seen glass bottles, you might have seen amber colored bottles, you might have seen translucent bottles. So there's several packing systems which help stabilize the molecule or the formulation. So in these cases, you might be just seeing the output, like they are giving it in a HDP form or they're giving in a blister form. I need to cut it open and take it. But there is a lot of research which is done in selection of a packing material, because that is the one which is reaching to the end user, and that is the pack which is for all the purpose, like the aesthetic which is required as part of patient compliance, as well as for the best route which it can be given. It needs to be stabilized in the tank. It needs to be convenient for a patient to be taken. So all these factors are considered. So even the now AI tools are helping us in identifying the packing selection. Okay. And the manufacturing robustness, like at small scale, we manufacture a 1 kg or a 2 kg dry powders or solutions of 1 kg, 2 kg. Now, when we scale up them, we take them up to 120 liters, or depending upon the requirements and depending upon the marketing requirements, we scale up. So it goes from 1x to almost 10x, 20x also. So when it goes such huge scale, there would be a lot of pharmaceutical manufacturing challenges which we face. There, AI helps us or AI guides us, okay, in taking decisions, quick decisions. So this is one of the software which I would just go through in more detail. And I also have told you, I have a disclaimer, all these softwares are used across the industry, and I've taken this information from public domain. Actually, Dr. Mastan has asked me to run you through the practical session, which I think it is not practically possible for me, because all these are highly costly and they don't allow us, and we have some limitations on showing information in the public domain also. So this information is taken from their public domain, but still I've used that, so I have some knowledge which I can at least tell you orally. So what does this software or the which is utilizing the artificial as well as the machine learning? It is used in drug compatibility studies, drug excipient compatibility studies. Here, like automatically, 800 plus excipients are used for screening for stability and solubility. 800 excipients are there in their database. Imagine if a formulator had to screen 800 excipients for his particular molecule, it might take more than 5 years, 6 years, frankly to say. But we have timelines of only 1.5 years to bring a molecule at least to the scale up stage. So other thing, predict 50 plus formulations. So these are all what they are doing is that there are two things they are doing. Majorly, they are reducing the time of development, as well as second thing is that they are decreasing the cost of development. In return, already morning I told you, cost is like the amount spent versus time. Suppose if you are going to take the development up to 5 years, we are out of the race in the market. So if you're 5 years, if you're taking to develop a product, and if you're out of the race, there would be a lot of people who might be utilizing AI, or who might be utilizing techniques such a way that they can come quickly into market. Once, suppose there is a 100 rupees market for a molecule, and there is one player will be getting that 100% of the share of the market. And if we are delaying our timelines, and if we are delaying our research, and we are late into the industry, late into the market, if 2% enter it, that 100% divided by 3 becomes 33.3%. So a company would be at loss of more than 50% of the share. So considering all these things, so we need to be quick, we need to spend some money, we need to come quickly into the market, as well as take the risk of using the current updated information available. Okay. Then excipient optimization studies, they also help us in generating impurities. Impurities generally, they are generated majorly on the stability, which might take about 6 months. So if you are going to develop a formulation, and at the end of 6th month, as per ICH guidelines, if you're going to see at 40-75, which is the accelerated condition, 40 degrees centigrade, 75% RH, it would take 6 months to identify what are the impurities which are coming up. Generally, what happens is that at the end of 3rd month also, if you're confident on your product, and if no impurities are generated, but they might come up or they might pop up at the end of the 6th month. So at the end of 6th month in your formulation, if there are some impurities which are being generated, then you would be at least loss of 6 months. So you should go back again, do the development. Now what the AI is doing is that it is helping predicting that there would be certain impurities which might be generated. So avoid using this excipient, which has it is not saying on simple terms that avoid using. There is a lot of chemistry involved, there's a lot of thermodynamics involved, there is a lot of background data, there are a lot of algorithms which are kept into the system, which help us identify that based upon its chemical structure, based upon its free molecules. It is going to predict that with this excipient, there are major challenges, and it will only predict. It is not going to assure us. So if you have 800 excipients, you have a choice to leave that excipient and do the development with the excipient, which is having less amount of incompatibility with that API. And suitable packing, I already gave you some brief information. So what are its key features? So it helps us in screening excipients. It helps us in giving virtual formulas. So it can screen n number of formulations with n number of excipients, and it gives us virtual formulas. Like it says that with this drug, you use these excipients. And it is again human intelligence which is required to assess the information given. The impurities which are being generated needs to be theoretically assessed. Okay. And the formulation stability, which I already spoke on. The impurity control, like control is by using the excipients which are having less exposure, and the drug-drug compatibility, and even the drug excipient compatibility. See, all these above steps, if I require to do a development, it would be taking for me to 3 to 4 years. But here, all the above steps, that's why I highlighted that all the above only done virtually. So virtually, why they are done? Because it is with the help of AI that we are able to virtually identify which excipient is having incompatibility, which formulation is generating more impurities, which combinations are generating, which packing material which is generating these impurities. And this is again a summary, like we can screen, this is again a number of solvents, like it is also used in preclinical or pre-assessment and all these things. Like we can screen a number of solvents. It is their advantages which they have put on the public domain. So they can, so one thing highlighted, they can evaluate up to 11,000 solvent systems. So which is slightly impossible for a human to use. We might not be requiring 11,000 solvents in our research development, but at least we might be using 15 to 20. At least using those 15 to 20 to evaluate which solvent approach is required, it might be taking at least 2 months. Okay. Now here is putting that they can evaluate up to 11,000 solvent systems. It might be a pure solvent single phase system, or a binary system where there is a combination of 2 solvents, or they can do a ternary system also. So these are one things like which are helping us, like precise development. Earlier was a development wherein at the end of 3rd month and 6th month we come to see impurities, and our heart is broken, saying that we lost the time and all these things, lost time, money, and a lot of stress. Now the AI is helping us in screening all those things. And the prediction are delivered between 8 to 12 hours within 1 day. Where are 6 months? Where is 1 day, 8 to 12 hours? So that to 8 to 12 hours, they take time to generate the report and share us the report. If we say that Actually, when I spoke to them, it takes 1 hour, it seems. 1 hour, and next half an hour, they can at least give us the tentative data that these excipients are having incompatibility with this drug. So where is 1 hour or 1 to 2 hours, and where is 6 months? So here itself you see a clear shift of development timelines, the money involved, and all these things. It might take few lakhs, but again, practically, if you are going to use the active pharmaceutical ingredient, they are very costly. Some are like almost like few thousands of dollars per gram. Now, the research majorly is into the oncology molecules and all these things. These oncology molecules, what happens is there are 2-3 risks with that. The cost of the molecule, it's very high. Second thing is handling the molecules. You know that oncology molecules, we need to take a lot of EHS. EHS means environment, health, safety. Risk assessment should be done. Personal risk assessment should be done. We cannot be directly exposed to these molecules. Okay, all these risks are there, and wherein the AI tools or these softwares using AI tools are helping us screen these excipients along with these APIs in a very short time. This is one practical example, like, but shown on theory, like what actually does the FarmDem AI or machine learning powered software does. So what does it do is that the excipient info, excipient information is not just the name. So, but we feed the name there. Practically what happens, I'm just trying to help you. Please visualize it, which I cannot show you on the screen. What does it do is when we feed one excipient, suppose you might be knowing microcrystalline cellulose, at least the final year students, you might have done in your industrial farms and all these things. Microcrystalline cellulose, when I feed microcrystalline cellulose into the software, it gives its entire data, like the structure, the molecular weight, what is its chemically content, it is the cellulose, what has happened into the cellulose, why it is microcrystalline, it's in a crystalline form, micro, it has been particularly size reduced, and all these things, aspects, everything will come. That in combination with the in energy terms, like the thermodynamic principles will be applied, wherein the heat and all these things will be applied in combination with the API. Like here the quantum mechanics and molecular mechanics are implied here. The quantum mechanics and the molecular mechanics of the molecular properties, as well as the chemistry rules. I might not be able to tell you what are the chemistry rules, but at least the chemistry staff can explain, like what happens during the what happens during the thermodynamics, like when it reacts with one of the drug molecules, what happened. Okay, what does it do is that the API and the excipient, when they interact, it takes all these into consideration. The stability, its molecular properties, its chemical nature, what happens on heat, what happens on exposure to the certain environmental conditions, like the relative humidity and all these things, and it gives the 3 aspects. Like if you see in the green zone, which is the low risk, followed by high risk and medium risk. So how does this come? This comes as a stability score, which is called the SS score. It is a stability score, which is an output parameter. So it gives in numerical form, like it gives negative, a neutral one, which is a 0, followed by a positive one. So here is that higher the positive, stability score is good, 0 is optimal medium risk, and negative is risk or high. I'll give you an example. I didn't name the excipients or the drug, but just I gave you an example wherein you can see that the values are ranging from minus 2.123 to 2.345. So here I've given you an example to A to K. These are the list. A denotes the excipient, B, C, D, and all this denotes the excipient. And when we feed each excipient in combination of drug into the software, it gives the SS values. The SS values are the stability scores values. And practically, I have seen that I have completed development using this software, wherein I saved a lot of time. What happens is that the molecule is a large molecule, and when used in combination with these excipients, it is having high degradation to oxidation. It is peroxide degradation is there, it is acidic degradation is there, basic degradation is there. So all these have led us to approach the PharmaDem to help us in identifying the compatibility. So using that, almost we got the scores like this only. Just these are all theoretical or fed just to keep you to help you understand. But if you see that the scores are like 2.123, 2.113, it doesn't mean that it means that in reverse, if I say, it means that B is having more compatibility than A. So if we come down the order, what happens is that this is only giving us the impurity generation or stabilization of the molecule with the drug, but there are other parameters which I need to take into consideration. This is only in terms of stability. If it is a liquid molecule and if I need to manufacture a clear solution or an oral solution, it should be clear. It doesn't mean that K, which is having a stability score of 2.435, which is most stable or most compatible with the drug molecule, I might not be able to use it because it creates incompatibility due to physical mixture. What happens is that when I mix an oil and a high HLB surfactant, what are they both separate? So the K molecule might be that it is having good stability score, but I cannot use it. So you can easily say that what is the challenge in your development? You can directly pick up K and J and you can do the development and you can complete it within 1 week to 10 days. No, it's not that easy actually. Artificial intelligence only guides us. So it is guiding me to use K molecule, which is having a stability score of 2.435. But with K, there are a lot of other challenges. It might be a physical challenge wherein it is incompatible, or it might be separating out on stability. Separating out physical compatibility is not shown here. Here it is showing only the chemical incompatibility by this score. So this is like, that's why AI is not the complete solution. Our human intelligence is a complete solution. So you should use this AI to go ahead and put complete efforts using your human intelligence, which I'll be speaking later also on the advantages and disadvantages also. So this is one example where we have used and we have come out successful and we have completed within, I think, 2 weeks or so. Actually, if I need to do a development with this, without using this software, it would take more than 6 months for me. And I need to put n number of formulations of stability. I need to have analytical work done on that. Without all these things also, we have done it quickly. And this is one more area where we are using this LASA limited Zenith software. What happens here is that I've taken an example of ranolazine wherein the API structure, we feed it into the system. Once we feed it into the system, and there is one more option called that at what temperature you want to run the study such way that you want to stress the molecule to understand. So this is somewhere called as post degradation. So post degradation, generally an analytical expert would take few days to months to do it. Okay, and he is limited to using limited excipients and all these things. But here, if you see that in the oxidation basic acidic peroxide, we can keep on increasing the degradation conditions. How many you want, you can put. And the temperature also 40 degrees, 30 degrees, 25 degrees. This literally will take less than 5 minutes. And if if an analytical scientist want to do, he will take more than 3 months to 4 months doing all these. Again, there are challenges in the analytical methods. Again, he needs to screen those analytical methods. He need to purchase a lot of things and all these things. So what does this? So at certain temperature condition and at certain degradation condition like the oxidation peroxide or acidic or whatever it might be, based upon its chemical structure, it predicts that there are 99% chances that such an impurity can be formed. And it will give an order like 96%, 95%, 90%. So seeing all this, at least we can pick up the top 4 impurities which might be generated. And by knowing the nature of the degradant form, like it's due to oxidative conditions, now a formulator needs to use his human intelligence and understand that I need to use an antioxidant in the formulation such a way that overcome this oxidation effect. Or if there is a basic degradation, he needs to play with the pH. So the pH on the basic side, if there is a degradation, then he needs to pull the formulation to a acidic side or a neutral side such way that that degradant is not formed. The next is on the acidic and peroxide. So whatever it might be, so at least it is indicating to you that after 6 months you will be knowing that I need to use an antioxidant, and that to usage of antioxidant, again you can use this to understand. So you can use n number of antioxidants. There are not no major antioxidants in the industry which we use. With this 10 to 15, using understanding first this, like what degradation the molecule tends to undergo, you can use this software and you can be able to know that which of my antioxidants is helping me to reduce the instability of the drug. And this is one diagram like which has the artificial neural network. Means what, we feed good amount of data into this, and once we feed the good amount of data, it gives a design space where we can use those excipients, like we say plus or minus 2%, 3%, and we take experiments at 2% and 4%, and we create a design space and understand that within this range of excipient use, or within this range of the excipients used in the formulation, you can be able to get good formulation. And this is also used in predicting the in vivo performance. Like we see here that in that particular paper, he has used this ANN network and he has created a design space and he has made formulations with that. And with that formulations, he has got good fasting and physical study. This is sufficient enough to file your molecule. Okay. And coming to the next topic, like the quality and compliance. In quality and compliance, how AI really works is that they detect the flaws, irregularities, and deviations very quickly. So how does this do? So what does it do? It improves quality control and decision making process. Like quality control means what? I have an example next. I'll show it to you. And it has enhanced real time visual impact and monitoring of products. So it does online. Generally, in the earlier days, what they used to do is that they used to pull out the batch which is being manufactured, and they used to manually see the defects and all these things. Now, but now what AI helps is that it helps us to do online verification and all these things. I have an example I'll show you. And enhanced manufacturing operations by optimizing the process and reducing waste. Okay. And also it helps streamlines tasks, minimizes mistakes, and guarantees compliance with regulatory standards. And it also enables ongoing enhancements and optimization. This is a simple example which I show you here. What is a simple film coated tablets? And you know there are a lot of defects in the film coated tablets, like twinning or the coating layer being removed, or there is rough surface. All these things you might be seeing in a Lackman book or in a general knowledge you have, that what are the defects of a film coated tablet. Now, what does this actually do? Like it has a digital camera which is actually connected to the ANN. That is, the algorithm is given saying that this is practically they feed it into the information saying that this defect it needs to pull out, it needs to separate, or it needs to separate the film coated tablet from the good tablets. And here he has given an example of diameter measurement. Like the diameter measurement is nothing but the thickness of the coating solution, coating onto a tablet. So it will be as precise as 0.2 mm or 0.3 mm. Coating is not going to add more than 0.2 mm to 0.3 mm thickness on your coating tablet. Suppose you have that parameter wherein you want to separate those good tablets with that of defect tablets. You feed all the information, and what does it do is that it classifies film coated tablets and it gives the thickness measurement, giving a standard graph saying that there is the deviation or the relative standard deviation between the actual value. Suppose your target is 15 mm plus minus 0.2 MFQ, that is 14.8 to 15.2. Which tablets are most deviating? Like we have a specification for that, and beyond that specification, the tablets are going to be rejected. So in such cases, this is an example of only diameter. You have other examples like the coating defects, wherever it might be, the compression coated tablet defects and all these things, which the AI algorithms are helping. Next is on the regulatory aspects, where in the regulatory department, this AI is clearly helping us. So it extracts a lot of data. So this is now, if you see AI usage in the pharmaceutical field with respect to the literature and all these things, I think it is majorly to be required in the area of clinical research as well as for the regulatory people, because regulatory people are those people who handle huge amount of documents, right, varying from thousands to thousands. Okay, so what they do is that they compile all the documents and they file to the regulatory authorities. So it is helping them in identifying the dossier, in helping in them in dossier compilation as well as dossier completion, and also in the internal audits and also data filing. So what are the softwares used here? Like here again, Ceratara AI is being used, wherein it gives accelerated regulatory and medical writing. Here the Ceratara AI is a larger AI. Under that, what is used for regulatory is the Gen AI co-author. So this co-author, like, it has several advantages. It helps in build transparency, consistency. It is a collaboration tool which reduces the submission timelines and all these things. It also helps generate documents such as patient narratives, clinical study reports, and all these things. So clinical study reports are huge documents. And the last is in the supply chain management. So this was also strange how AI is being used in supply chain management. In supply chain management, what does it do is that they take the algorithms and they fit it to the transport vehicles. So suppose if a drug molecule is manufactured in Guntur and it needs to go to several parts of the world like Europe, Asia, different parts of Asia and all this, what they do is that they fit the algorithms and they try to understand how it is being. That is, it is simple say that it is a management. How it is here AI tool is used to manage the shipments. Okay, it's not only in research, it is being used in several areas of the industry. Okay. And to summarize on the same, like AI can analyze vast data, which I have already told. They identify patterns, they make predictions, they improve the efficiency and effectiveness of drug development process. Also, it has been helpful in the data which comes up. It helps us in making decision making, which is very important for a scientist. So making decision is important. Making decision today, you take a decision. After 6 months, we are going to take a decision. It's of no use. So it is helping us take decision quickly. That is important. And also like giving personalized medicines. So based upon your gene therapy and based upon your diseased condition, based upon your adverse events or based upon your symptoms, now there is development going on to target a particular patient. So the oncology molecules, what they do is that when you take an oncology molecule, it has several adverse events and several side effects due to which the cancer patient, the more benefits which are to be which are to be driven through the medication which is taking, the adverse events are killing the patient. So what is happening is that they even kill the good cells. Now in the oncology department, like using the AI and all, they're developing a lot of targeted gene therapies and all these things. They give a personalized medication. Okay. And a general statement is AI and ML, which is very important. It is not replacing formulation scientists or process engineers. What does it do is that it is just amplifying their expertise. Okay. Now challenges. There are a lot of challenges which I can say that is the knowledge and experience of human researchers cannot be replaced by AI. Okay. And even though theoretically it is helping us know many of the formulations and all these things, but practically you need to do minimal experimentation. Like it cannot replace. Why I'm giving a statement is that AI can never replace human intelligence. Okay. It is in few areas wherein it is gaining importance, but this is a current day statement. But after a few, if there is more research going on AI and if it is going to come up, but as on current date, the myth which is going outside is that is AI is replacing a lot of jobs and all these things. But at least in the science field, pharma field, or in the areas of medicinal research and all these things, it is only helping or amplifying. It cannot replace. Okay. And data collected. So there will be huge data, and if it is again fed by humans. When you feed the data, if a human is making an error, your AI will not be able to make that. So it takes the data as such, and if the data fed is not right, it is going to give you wrong summary. So all these challenges are there. So it is a combination of AI and HI, which will help the clear drug development or pharmaceutical. It will help the pharmaceutical industry. There are a lot of ethical concerns, social concerns, and all these things. And finally, trans versus truth. So artificial intelligence versus human intelligence. Still, I believe till date, today, as on date, human intelligence is more efficient than artificial intelligence. Only that artificial intelligence just helps us or amplify or reduces time or reduces money in the area of development. Thank you. And if you have any questions, I would be happy to answer, provided I have that answers. If not, at least I can help you in the area where you can go and get these answers. Thank you once again. I'm happy to take questions if there are any. Yes.
The display camera you are saying to capture it. And after capturing the image process to the YouTube format. So this how to convert this image into pixels? If there is some AI tool or something manually we calculate the image pixels, how is this process with the YouTube format?
Okay. So I may not be able to help you with the background what is happening, but practically what happens is there will be a conveyor belt, and there would be a camera fixed focusing on the camera belt, on the conveyor belt. What does it do is that there is already information fed into the camera pattern using this AN and algorithms that such and such thickness is my specification. So, and the conveyor belt will be given an algorithm good tablets basis the weight and all these things. Okay, the same phenomenon here that the information which you feed the, this is again you're feeding the data to the by creating AN or the algorithms which you are giving a output to the camera and input to the camera, and the camera detects. So it is very precise that, that's why I was telling you that the coating thickness would be 0.1 mm to 0.2 mm, 0.3 mm, which is difficult for a human naked eye to identify, and he needs to measure the 1 lakh tablets physically to understand it. But here what does the camera does is that it takes the images, it reads the images, it helps knowing that the thickness is high or low, automatically separates those tablets which are defective. So this is one example, but there are a lot of areas where it helps us that in identifying the pinholes, in identifying the coating defects and all these things. Yeah.
So I think we all have coding, right? The data. Just to align this on what you have asked. I think the data is all very good based on the data that we given. Matter whatever size are minus market, like input all the data and we probably thousands of images showing a compare it. So if you matching it, then it's going to take it as
data what you are going to give to the artificial AI is more important.
Yes, sir. Yes, sir.
That is not.
So the more the data, the prediction. So, this note, I thank you, sir. Sunil, sir, for this beautiful session which you have engaged our students and as well as other faculty members. We thank you on behalf of Archari Nagarjuna University College of Pharmaceutical Sciences, and I also thank the faculty members for the important session through the online mode. So I thank each and every one of you for your cooperation. I also thank all the participants who have joined us through our YouTube channel. We look forward for the same engagement and participation and cooperation from all of you throughout this week, where we are going to continue our FDP sessions. And now I request all of you to join for the lunch, and later there will be 2 more sessions. Okay, thank you very much.